All right, well, thank you guys for having me. I'm really excited to talk about this. We actually just had to reanalyze some of my data because we got some more samples and stuff. So you guys are actually seeing some new data that we just got, which is exciting. Like Matthew had said, that's going to be a little bit about what we're doing overall in the Phillips Lab at the University of Southern Mississippi. And then I'm going to talk mostly about what I'm doing with Largetooth Sawfish and what we're looking at for my PhD. So just a brief overview. So I kind of had an introduction, but I'll tell you a little bit more about who I am and how I kind of got into this. I'm from Florida, and I went to University of Florida as an undergrad. And during that time, I was working at the museum there, and I was working at the Florida program for shark research. So you can see me here with a sawfish saw before I even got into grad school and actually started working with sawfish. And during this time, I was actually helping out with, we have an encounter database for sawfish encounters. And so what we were doing is anyone who saw a sawfish would actually call us at the museum and tell us, where they saw the sawfish, what they were using, if they were fishing, things like that, or if they saw them diving. And here in this video, you guys can actually see a sawfish if you've never seen one before. And I'll kind of give an overview of what they are. But you can see here they have that long saw, and that's what they're really named for. And they mostly use that for finding food. And they are raised, so their mouth is on the bottom, and so are their gills. And so you can see here, this sawfish, it'll kind of pin a fish to the ground, and then it sucks the fish up with its little mouth. And these guys can get really big. A lot of people don't realize that. They can get up to about 23 feet, so pretty big. And we do have some here in the US. So again, in Florida, that's where we have one of the species. It's one of the last places you can find one of the species of sawfish. So that's why we have the encounter database there. And while I was working there, I actually met my professor, Dr. Nicole Phillips. And so this is her. I don't know if you can see my, let me see if I can find a spotlight. That's Nicole. So she actually came to the museum to sample some of the saws that we had there. And that's how I kind of got into this. So I've been at USM since 2016. So I did my master's here, doing the same work. And now I'm doing my PhD in it. So once you kind of get into sawfish science, it's really hard to get out of it. You kind of get addicted in a sense. So yeah, I've been working on them now for, since like 2015 or so. So a long time. But anyways, that's kind of how I got into this. So now I'm going to kind of go into what my project is and what we're really looking at with these saws, and really just museum specimens in general and why they're so important. So everyone's probably aware of biodiversity. It's kind of a big term now that everyone's using. And there's really 4 different levels to it that's important. And one of the ones that we're really focusing on with our studies is genetic diversity. So it's kind of the basis level of biodiversity. And so it's really important for populations and species to have genetic diversity. And if it's lost, what can happen is that can really cascade through all the levels of biodiversity. And it can end up, you can lose a whole ecosystem if you start losing species and their genetic diversity. So it's really important to understand where the genetic diversity is for a species if you are trying to preserve that species. Now, right now, a lot of people, again, might be aware that biodiversity is declining. So right now, we've lost about 68% of populations from vertebrates. So this is kind of a graph that's showing that. Again, these are just vertebrate populations that we've seen. But a lot of people know with the large mammals that we've lost. Amphibians is a huge one, like all herps are not doing so well. And then chondrichthyans, so those sharks and rays. We're losing a lot of those populations. And again, a lot of people think of biodiversity and like extinctions or species extinctions, but a lot of people don't think of populations and what populations we've lost. So that's one thing you can look at when you're looking at genetic diversity is those population losses and what those actually mean before that species actually goes extinct. And so that's really what we're kind of trying to focus on, is what have we lost in terms of biodiversity. So one thing you can look at when you're looking at it, again, is what I was talking about, is population. So the spatial patterns of genetic diversity. If you have a population like we do with Largetooth Sawfish that's found all around the world, how genetically different are those populations? You know, are they all one population, they're all interbreeding, or are they all genetically distinct and not interbreeding? And this is really important to know for management. You know, if you're going to manage a population, do you need to do it all as one, or do you need to focus on certain areas differently? So that's one thing that you can look at with genetic diversity. Another big thing is temporal patterns. So how has this genetic diversity changed over time? And again, if we've lost a lot of biodiversity, that means we've probably lost a lot of individuals. And so those individuals were not able to pass on their genes. And especially if those genes were unique, that means that you've lost some of that unique genetic diversity. So what you can also do with genetic diversity is compare it. So you can use museum specimens to compare to measures of genetic diversity today. Like if you go out and catch a sawfish or a shark or something and take a genetic sample from it. So I kind of got ahead of myself there with the museum specimens, but it's hard to determine spatial patterns and temporal patterns if you don't have assessments from back in the day of genetic diversity. And again, DNA wasn't really found until the late 20th century. So people weren't out there taking genetic samples or samples that were preserved for genetic use back in the day. So we don't really have a good idea of what the genetic diversity was like for many species, in the early 1900s or before then. So before they've experienced all these declines over, the 1900s, we don't know what their genetic diversity was like. And what we do know now is from populations that have already seen large declines. So we don't know, are these populations, is this what their genetic diversity has always been like? Is it really low now? We have no idea. And so this is where museums come in. This is what makes them so important, is it helps us to go back in time and look at these populations and their genetics. So we can actually go back and look at populations from the 1900s or from earlier. Some museums have specimens that, are from the 1600s. And you can actually assess the genetic diversity that was found then and compare it to genetic diversity today to see how it's changed. And we would have no way of doing this if it weren't for museum specimens. So a lot of people think, museum specimens are just for looking at. They're not. They're very, very important. Now, the one thing about museum specimens is, like you see here, a lot of these are in jars, and a lot of these are in ethanol. Many museum specimens were formalin fixed back in the day, so they were put in a formalin preservative. Usually that's to help with like keeping color and morphological characteristics. But what it does, it actually kind of messes up the DNA, where it's really hard to get it out after it's been formalin fixed. So for a lot of museum specimens, it's really difficult to actually get DNA out of them. And some people have actually figured out ways that you can do this. But again, it all depends on the specimen. So for many specimens, you need really certain kinds of specimens. Ones that are usually dried are the best. And so this is where sawfish comes in. Sawfish kind of gives us this unique opportunity to go back with a shark species and a ray species to look at their historic populations because they're dried. So they, a lot of times, were not formalin fixed. These specimens have been dried, and therefore the DNA is better preserved for us to use. Again, a lot of shark species, like you saw here, a lot of them are formalin fixed. So sawfish are this unique kind of group that we can actually use to assess these things for a shark or ray species. And this hasn't been done yet. So with really with chondrichthyans, we do have a big gap in our understanding of how much of them we've lost, what we've lost in terms of their genetic diversity. So sawfish provide this really awesome opportunity to do this. And you can see here, this is a largetooth sawfish rostrum. And this is my species of study. And so these guys are critically endangered on the IUCN. So that means they are on the brink of extinction. Now, all sawfish species, there's 5 different species. They're all either listed as critically endangered or endangered. So all of them aren't doing great in terms of their extinction risk. And these guys are one of the biggest species. Again, these ones are the ones that get up to 23 feet. And right now, one of the last places you can find them is in Australia. Now, you might be wondering, how did we end up with all of these saws? Well, sawfish are coastal. Usually they are found in areas that are heavily fished. So they do come in contact with fishing gear. And this happened back in the day before they were protected. They would get entangled in this fishing gear. And you can see here, this is a largetooth on the left that's really heavily entangled. And so what people would do is they would actually cut the saw from the sawfish. And that does not grow back. And they use that for finding food. So they actually use that saw it has electrosensors on there to help them find fish. If they're in really muddy water, they can actually sense the fish. And they'll stun the fish using their saw. And then they can run over and eat it. Without that saw, it's really hard for them to find food. So what ends up happening, they found, is they'll end up starving to death. So this is what happened back in the day a lot, which is rather a grim story, but sawfish, again, lost a lot of their saws from incidences like this. Now, this video here, if you don't want to see this, this is someone who is attacking a sawfish. This, I believe, was from 2020. This was in Brazil. Now, they are still found in Brazil, but we don't know. Yeah, this is them. This is a fishing boat. They found a sawfish. This was a huge sawfish too. And they are actually protected in Brazil, but you can see, they're cutting the saw off there. So this is how we've ended up with so many saws now in museums all over the world. Is a lot of people would keep them as trophies or trade them. They can, like in Brazil, they actually use the teeth for cockfighting, which is interesting. They're actually thought to be better than like the metal spurs that they use. So they can be sold. I think a pair of them can be sold for like 80 to 100 US dollars. So people will sell them for things like that on black markets. And so although this happened a lot historically, it does still happen today. And even in the United States, they have, there was recently someone who I think was arrested for killing sawfish in Florida. So this isn't something that just happened historically. Now, for largetooth sawfish, they were found circumtropically. And so all these blue areas here on this map are where they used to be found. And today, this is their current range. Now, although a lot of these places you can see are still in blue, really the last place that's considered their global stronghold is Northern Australia. So that's where you can go and you can actually find them in reliably large numbers. So that's why we can consider it the stronghold. In some of these other areas, like again, I said in Brazil, you can still find them, but a lot of times it ends up like you saw in the video, they'll end up being killed. And we don't really have a good idea of really what their populations look like in these other areas. We just know that there are encounters still with them, but we don't know how many there are or anything. Now, there's 2 areas that we consider beacons of hope. So again, these are areas where we are still seeing encounters with them more frequently than these other areas in blue, besides Australia. So Brazil is one of the beacons of hope, and then Papua New Guinea is another beacon of hope as well. But again, in Papua New Guinea, I don't believe there's actually any protections for them. In Brazil, again, there are, but you saw that video, a lot of times there's just not enough enforcement to actually put those protections in place. So yeah, there's not really, Australia is kind of our last big place where we can still see them. Now, we do have some indication of their spatial patterns of genetic diversity. So someone actually did a study for their PhD a long time ago, looking at, how different they were over time, over their range, but they didn't have a lot of samples in there. So we do need to clarify this a little bit and get more samples from each location. And then my professor actually looked at contemporary patterns of genetic diversity in Australia. So she was actually the one to figure out how to actually get DNA out of these saws. And she did that for part of her PhD work. And then she also she gathered samples from sawfish that they caught in Australia in the wild, and then looked at their genetic diversity in Australia. And again, they're kind of all over the map. Some of the genetic studies say that it's low, and then some say it's high compared to other shark and ray species. So even though we do have these indications, we don't really have anything to compare it to. So we still don't know, are they good genetically? Are they not? Do they have low genetic diversity? Or do they have high genetic diversity? We really don't know. So that's kind of the big question, is we need to figure out where their genetic diversity is at. And then also, again, we can look at whether they have lost genetic diversity over time in Australia. If that is kind of our last global stronghold for the species, we can look at historically, did they have a lot more genetic diversity, so different genes, than we are seeing in populations today. So that's really what we're trying to focus on. So we're using these museum specimens to go back in time and answer these questions about, how different are these populations that we still have and we had historically, and then how have these levels of genetic diversity changed over time. And so we've been doing this now. We started this in 2017, and we've been going around and looking for these saws and all over. We have one from a woman who said she had one in her kitchen. So that's above her sink in the top left corner. This one is from the Smithsonian in the bottom left. On the bottom right, this is one of the largest saws we'd ever seen. They had a pile of them in the National Museum, Scotland. And then on the top right is a random bar in Australia that we found some in that were just on the wall. So they're kind of everywhere. There's actually one that I found recently in Hattiesburg, where we live now, in a lawnmower store. So they're really everywhere. And now that you've seen this, you'll probably start seeing them a lot more, because they don't just have to be in places where they still find sawfish. They're in the most random places. So we've dubbed this search for sawfish saws as Saw Search, #Saw Search. So if you want to go back and look at where we've been, you can find us on Twitter or on Instagram and look at the hashtag. But we do a lot of outreach. So like this for us, me talking to everyone here helps us in a way, because it helps us get the word out. And, one of you may know of where a sawfish saw is. So we do a lot of outreach with people, just getting the word out. And that also helps us find more sawfish saws. And it also helps tell people about sawfish. So that's another reason that we do like doing this with natural history museums and collections, because it shows people how you can actually use these collections. It's not just something that's cool to look at. These collections are very important for saving biodiversity. So in 2017, when I first started my master's, we decided to go to Florida and look for these. Now, again, Largetooth, they were thought to be found here, but they're not found in the US anymore. But again, that doesn't matter. You can find them really anywhere. So in Florida, we did find, I think, around 100 or so in Florida. This was one at a random bar that we went to. This is us. We don't look very happy. This is us fishing them out of the Smithsonian. This is like the end of our trip. And we were fishing them out of those large barrels of ethanol. So even though it's really hard to get DNA from ones that are in ethanol, because we don't know whether they've been formalin fixed, we do still take samples of them, because sometimes you can get the DNA out of them. Then in 2018, we actually went to the UK, and we went all over the UK. So all these little places on the map are places that had sawfish saws. So we actually didn't realize how many were in the UK. We ended up getting, I believe, over 500 from there. So they've never been found in the UK, but a lot of people from the UK have been other places historically. So they brought back some sawfish saws. And yeah, we ended up getting a ton from there. And even across the street from our Airbnb on the first day, there was a fish and chip shop. And you can see in the background, there was, let me see if I have my spotlight. There's a green sawfish saw hanging on the wall. And so we were just walking by it and saw it and went inside and we're like, hey, we came all the way here to find these. Can we sample it? And the owner was like, yeah, come back tomorrow morning. And he let us sample it. So it was a really successful trip. And then after the UK, I actually went to Australia to find some there. And then again, this is a very random, like a very small museum. It's called the Never Never Museum. It was in the middle of the Australian outback and it had the largest saw I've ever seen. It was from a 6.2 meter sawfish. So, and it was just on the ground of the collection, just sitting there. So we went around there and got a lot from personal collections. So for a lot from people who had them in their house, who had them for years before they were protected in Australia. And I also went out trying to find them in the wild, but even after 11 days, I still have never seen a sawfish in the wild. So they're that rare that I've gone out looking for sawfish 2 times now, spending, more than 10 days at a time and still can't find one. So just shows you how rare they are. Then again, the Natural History Society of Maryland, so you all had 6 saws in your collection. And I actually looked on Google as well. You can see in one of the photos, they're in like a glass case underneath, I think, a turtle shell. And then, yeah, they said some undergraduates from University of Howard helped collect information from them. So that was really awesome. So we are still collecting. So this happened this year. So we're always going to be collecting. My professor is going to be working on this, for her life. So this project is really never ending. I'm only doing a small part of it. I'm doing Largetooth, really until we'll still be collecting forever, but I'm just doing 1 species. There are 5. Someone else is doing smalltooth, and then we have 3 other species that needs to be done. So we're just collecting, waiting for students to come in and do this work. Now we have collaborators around the world. So we have the Shark Trust, who's helping us in the UK, who's still collecting for us. We have people in Sweden who are collecting. One of our collaborators in Northern Australia who's still collecting. We had someone go to Singapore and collect. And then Papua New Guinea, we're actually getting samples from a PhD student who did his work in Papua New Guinea, and he collected samples for us. So we have a lot of collaborators, and we couldn't do this without collaborations. Again, even, and like you all, I couldn't get up there and sample them myself. So you all generously sampled them for us and sent them to us, so that we can actually get information out of them. And it helps us a lot because we don't have the money to go and do all these trips. We got a grant to go the 1 year to the UK, in Australia, and then around the US. But now we really rely on people sampling them with our kits and then sending them back to us, which is really helpful. So overall, we've sampled from over 150 collections. We have over 1,500 from all 5 of the species. And just for my Largetooth, I now have probably around 350. And so if you guys have questions at all, like about that stuff, feel free to put it in the chat, or you can just turn off your mic and ask as well. But now I'll kind of go more into how we're actually doing the work. So I just want to take a minute and see if anyone had any questions about the saw search stuff. Okay. So now we're going to go into the genetics part, which again, I won't make it. I used to hate genetics before I actually started working on it. So I'm not going to make it very like class or anything. But I'll explain kind of how we are doing this work and how we're going in and actually sampling. So you can see here, let me get my spotlight back.
One question has come in.
Yeah.
Annmarie.
Where's the DNA? Oh, here we go. So perfect. I'll explain this now, actually. So my professor, when she was in school, she sampled from the teeth, from the side of the rostrum. Inside, there's like this little area here. It looks kind of like, I always call it like the skull area. That's not correct, anatomically correct, but it looks kind of like a skull, like bony area to me. And she found that this area here, right underneath the skin, so it's this white kind of tissue, is the best place to get DNA. So I think she got DNA out of some of the other places. The teeth were like really the worst. It really didn't get a lot, but this area gave the best amount of DNA. So that's where we sample from, is the base. And again, you can sample from really anywhere underneath the skin here. It's all cartilage. And what you're really looking for is this white tissue, gives the best DNA. And on some of these, I don't think you can really see it on here, but there's a tissue that looks kind of like a honeycomb tissue. And it's really hard. That stuff does not break down, so you can't really get DNA out of it. So that's why I try and tell people, like, sample this white, kind of fleshy looking stuff. But that's where we get the DNA out of. So that's what we're looking for when we go into museums. And we don't, like, I want to work in museums when I graduate. So I made it a point to not try and, destroy specimens. That's kind of always something that we do struggle with, is when you go in and try and collect from specimens. It's the curator's job or the collection manager's job to make sure you're not destroying the specimen. And what we're doing is we are destructively sampling, is what it's called. So we try and do it in a way that is aesthetically pleasing as we can. So we'll either sample from the underside if they are going to hang it up, or we'll sample along the edge where it's already cut. So you can't really tell. We had one museum who wanted us to show, like, they wanted us to take like a block of it, so they knew it was sampled, which was kind of cool. I thought that was interesting. But for the most part, when we do sample from these, we don't want it to look like it's been, hacked up. We want to save these specimens as much as we can. But again, you also want to get the DNA out of them because it does have so much information. Now, when we take these back to the lab, I'm just going to do a comparison of what you would see if you were to go and take a fin clip. So this is a fin clip from some kind of shark. And this is what it would look like. You have this giant thing of a fin. You get really, really great intact DNA from it. And you typically will run it on a gel, which will show you, like, the higher the band, the more DNA, the longer the DNA is, the better quality it is. And so this is what you'd look like if you were going out and collecting from sawfish today, like out in the wild. Now, this is what we get, is we get this really old tissue from museum specimens that have not been preserved for genetic use. They are dried, so they haven't been formalin fixed, so it is more intact, but it's still very broken down. So this is what my gels, it's actually a very good gel that I got, where we actually had bands. A lot of times my gels will be almost blank. You can't tell that it has any DNA. And so that's really the DNA we're working at. So it is really difficult to work with historic DNA, is what we call it. So it does take a lot of work to do, and you don't get DNA out of every specimen. Like for this specimen, this was one I did in Australia that I found in an aquarium there, actually had one, and it was really humid, and it was literally disintegrating, the saw. So sometimes, again, you're sampling from these. I still took a sample from it, but I mean, the teeth were disintegrating as I was touching it. So because these aren't specimens that are preserved for genetic use, you kind of, you don't really know what you'll get. And that's kind of the nature of working with museum specimens. On one hand, it's really fun and you know, it's always exciting, but on the other hand, you don't always get DNA out of things. And so a lot of you may have, if you've attended other, you know, conferences or talks, you may have seen people talking about genetic stuff. There's really two ways you can do the genetics of any kind of animal. You can do the mitochondrial DNA or the nuclear DNA. And I didn't know this until I started working with it. But the mitochondrial DNA, which is what we're doing right now, there's more copies of this in every single cell you have. And it just shows female patterns of genetic diversity. So it's passed down through the mother. But you have a ton of copies in each of your cells for mitochondrial DNA. For nuclear DNA, you only have 2 copies in each cell. But it does show the patterns from males and females. So whenever you're choosing what kind of DNA you want to do, you have to think about the questions you're trying to answer. Because we are working with really degraded DNA already, we wanted the best chance of getting DNA out of them. So we went with the more copies, which is mitochondrial. And typically, if you're working with degraded tissue, that's what people are going to work with, is mitochondrial DNA. And so we went with one of the regions, so it's just a part of the mitochondrial DNA. We did not do the whole genome. So our region that we're looking at is probably 1,000 or so base pairs long. And so once I've extracted the DNA, what we do is we do a polymerase chain reaction. So we just make a ton of copies of that DNA. And we can do it on a regular PCR machine, which you can see here at the top is this gel here. Or we do also a more sensitive version of PCR called a quantitative real-time PCR. And it's just more sensitive. It can detect down to 2 copies of DNA in any sample. So that's really helpful, again, because the quality of our DNA is not great. So once I extract the DNA, amplify it, I then send it off. We don't actually do the sequencing here. We send it off to a company who will sequence our DNA, and we get back something that looks like this. So each of the base pairs from our samples. And just a reminder, if you hadn't had a genetics class in a while, the DNA is made of A's, G's, C's, and T's. And so we're just looking at the different patterns they make up. And so what I'm looking at when I'm looking at all my samples, I'll kind of line them up and I'll look for differences in the base pairs. So on one hand, some samples from Africa may have a T, and others from, say Brazil might have a C here instead. And what that means is you have 2 different haplotypes. So this is how we look at genetic differences between all of our samples. And we can see, are populations genetically different or are they genetically similar? And so what we looked at, this is just some preliminary data. This is the new data we have. So our sample sizes are bigger, but I got a really big fragment, about 800 base pairs that I analyzed about a month ago, just to kind of see what we were getting. And so I have broken them up into how they're broken up by the IUCN. And so they have an Eastern Pacific, a Western Atlantic, Eastern Atlantic, and Indo-West Pacific region for this species. And so these are my sample sizes from all of them. And I looked at the genetics of each of these regions to see, are they different or are they similar? What do they look like? And so what we saw, I ended up finding out of this 800 base pairs, it was about, I believe, around 57 samples. So out of those 300, if you can imagine, 57 of them so far have given me 800 base pairs. So again, it doesn't seem like a lot, but a lot of those 300 samples may have been ethanol preserved or formalin fixed. Some of them just didn't have all the sections that we needed. So out of those right now, we have 57 that have that full 800 base pairs that work for us. And so this is what we're seeing. We got 23 haplotypes, so 23 genetic differences that I saw. And each of these, I'll kind of walk you through what we're looking at here. So again, up here are the haplotypes. And let me get here we go. So these are the haplotypes, each of these different colors. And each of these circles represents like the population that's down there, or the samples from that region. And so there's different colors represent the haplotypes that are found there. And the amount of the pie chart that it makes up is the number of samples that had that haplotype. So for haplotype 1, it makes up most of the Africa samples we're seeing, is this haplotype 1. And then a lot of them were haplotype 2. Now you'll notice most of these are like haplotypes that are only found in these regions. So it's not, we think that each of these are probably genetically distinct in a way. And the size of the whole pie chart represents how many samples we had from each of those regions. So we're looking for more Eastern Pacific samples right now. That's where we have the lowest sample number. Eastern Atlantic, we actually got a ton from someone who did their PhD work in Gambia. So they took about, I want to say almost 200 samples from sawfish there, and half of those were Largetooth. So we got a lot of samples from East Africa, which are great. And the biggest thing that came out of this and what we saw is that these two regions are different. So the Eastern Atlantic, Western Atlantic, and Eastern Pacific are a different lineage than the Indo-West Pacific is what we're seeing right now. Now, this is, I'll kind of explain in a bit why this is such a big deal for conservation. But this is kind of what we were trying to see with the spatial patterns is, is this one big population? No, we don't think so. It's definitely two different lineages of Largetooth sawfish. And then what we wanted to see again is how much of that genetic diversity have we lost? If we're considering the last global stronghold to be in Australia, so if discounting all of the other tiny populations that you might find around the world still, really Australia is kind of the last place that we're thinking these might survive. So if that's true, how much genetic diversity have we lost? And so far we're seeing almost 80% of that genetic diversity is lost if we lose all of those unique genetic populations. Again, as you saw in the map earlier, a lot of them have already been lost, which is not good. But yeah, so this is what we're seeing that still remains in Australia. So it's only 4 right now of what we saw historically. And again, this is only 57 samples. So we might see more as we add more samples in.
Question in the chat, Annmarie.
Oh, yeah. What type of proteins are encoded in that piece of DNA? Oh, so for the region that I'm looking at, it actually doesn't code proteins. So it's a non-coding region. And we chose that because it typically has the most mutations. So you'll see the most differences between populations there. If you choose a region that is coding, so it's coding for proteins, and you're looking within one species, that might actually all be the same, because typically coding regions are very I'm losing my word here, but they're all typically the same because they're all coding for the same protein that species has. So if you're looking within, highly conserved, there we go. So yeah, they're usually conserved. So we went for a non-conserved region, and the coding region is, or the control region is usually the one that has the most mutations. Okay. And so then we also did this just within Australia. So a lot of our Australian samples are only reaching back right now into the 70s. But what we've seen since then, with our historic Australia samples and the contemporary samples that have been done, is that there hasn't been a decline genetically, which is good news. That's good news for Australia. But yeah, right now we saw all of the historic haplotypes we saw. We're also seeing in the historic studies, which is good news. Now, in terms of conservation, what does this really mean? So within Australia, if that's our global stronghold, what do we need to be doing there? You know, what should we be focusing on? And really, if we haven't lost a lot of that genetic diversity, we just really need to keep maintaining those populations over time. If right now the Northern Territory and Western Australia where they're found, there's not, like when I was out there, there's not a lot of people out there. So that's why we think, you know, a lot of these places are still pretty pristine, which is probably why they're still found there. It's really hard to get out there and fish for them. But they are still caught in like some of the shrimp trawling and stuff in Australia. So people are working in Australia to help people understand how to get them out of nets and make sure that they aren't killing them there. And that's really the biggest thing, is making sure we don't lose any more of them. Because once you start losing more of those individuals, you'll lose more genetic diversity. So that's really what we should probably focus on in Australia. But then outside of Australia, again, we still have some of those populations. So what do we need to focus on if it's not in the global stronghold? And really, Brazil is where we need to work, or where we need to look at. So that's part of that unique lineage. You know, if it's part of this one over here, I only get my spotlight. So if this is a distinct lineage right now, if we kind of ignore it and just focus on Australia, we're losing all of that genetic diversity that might be unique and is unique from Australia. And it's still, Brazil is one of those areas that's considered a beacon of hope. So if we can work in Brazil in maintaining sawfish there, we can maintain some of that genetic diversity. So what we're really working on, I still have probably 2 more years in my PhD. If I can get out in 2 years, we'll see. But right now we're trying to get more samples from Brazil and Papua New Guinea. We have, I think, 100 from Papua New Guinea right now sitting in Australia that we're waiting to ship over. And then Brazil's a tricky one, because right now you can't get samples out of Brazil. This is a CITES listed species. And Brazil at the moment wants work to be done there. So I've actually put in a grant if we can go to Brazil and do all of the work there. We have about, I want to say around 100 that are sitting in Brazil that I could go and sequence, which would be really awesome, because right now we don't know anything about them genetically there. So that would be amazing to be able to go there and look at what's going on in Brazil, and even potentially do some comparisons within Brazil and see how much genetic diversity, if any, they've lost. Another big question in Largetooth sawfish is Papua New Guinea and Australia, and whether they're one population. And in Papua New Guinea, they are still fished, and they get caught a lot in fishing gear and stuff, and they're not protected. So if it is one population, if they're getting caught in Papua New Guinea and killed, it is affecting that global stronghold. So we need to figure out, are they connected genetically or no? Because that definitely helps determine how we should go about their conservation. And then another really cool thing that I'm excited about is we're going to do a population assignment testing. This is something that my professor has been wanting to do for a while, and you have to wait until you have a lot of samples for a species to be able to do this. But what we'll eventually be able to do once we have enough Largetooth, and again, hopefully we'll be able to do it at least for some populations of my PhD, but eventually we hopefully will be able to do it with each of the 4 regions they're found, is if a collection has samples or specimens, they don't know where they're from and they're Largetooth, we'll actually be able to genetically test what region they likely came from. So that'll be awesome, because then it'll open up that specimen to being used in even more studies, because a lot of times now, if it doesn't have collection location information, people won't use it. So this will be awesome for all the museums that we've been to. We'll be able to tell them, you know, this specimen is from Brazil, or this specimen is from likely East Atlantic, stuff like that. So that's something else that we are working towards. So I just want to thank everyone on my committee, all the volunteers. These are all my volunteers that have helped me find sawfish. I have them go into Google sometimes and just type in museums and look at their photos to see if they see any sawfish on the wall and stuff. And then all our collaborators who have helped sample with us. And then all the museums that we have been to, all the institutions. You can see you guys are on there as well. So thank you for everyone who has helped us sample. And then all the private collectors. This doesn't even include all the private collectors that have helped us, but they've been a huge help as well. So with that, I'll take any questions from anybody.
There's one in the chat that came in a short time ago about whether we know what's reducing the populations.
Yeah, so really all of this, we don't know anything about disease yet. That's one thing that can be affected. I'll stop sharing my share. So disease is one thing we don't know about, but if they have low genetic diversity in an area, it does make them more prone to disease. So that's kind of one thing that'll reveal itself once we figure out, you know, how much diversity they've lost. Climate change is another big one. They do use, so Largetooth in particular use freshwater. So they'll actually give birth at river mouths, and the influx of fresh water from a river will actually like help cue a mother to pup. And then those pups will live in the river for a couple of years before they actually go into an estuary and then the marine environment. And right now, one of the big things in Australia is they're trying to use those rivers for cattle. And so they're actually trying to pump out some of that water. So that's another thing, is like human degradation plus habitat degradation is really affecting them. But then I think exploitation's been one of the biggest, is just getting caught in fishing nets and people killing them. And so the next one is, how long you've been working on this? So I started working on sawfish in 2015. I've been in this lab since 2016, so a long time. So my master's, I ended my master's in 2020 and then started my PhD because I was like, well, I'm already working on sawfish. I like it a lot. And my professor was like, just keep doing what you're doing and we can do even more. So yeah, I've been working on it since, I guess, like 6-7 years now. Yeah, it is a lot of dedication, and it's so a long time, but it's fun. I really like it because it's such an interesting subject. Oh, thanks, Jeff. Jeff is the one I was telling you about that is like sharks and stuff, and he wanted to come. Okay, someone else did an 8. See, genetics is, okay, I've heard that genetics takes a long time. He said he took 8 years for genetics PhD. It takes such a long time because you have to go out and get all your samples, which is a lot of hard work. Do their teeth grow back? So if they're cut, like if a tooth is broken, it can still keep it'll keep growing. So they do continuously grow. But if they lose that tooth completely, it won't grow back.
Are there questions?
And if anyone knows of any saws anywhere, I believe you guys have my email. So please email us. I will
Every
try.
time we go into bars out on the Eastern Shore now, we're going to be like, where's the sawtooth on the wall? Totally.
Yeah, no, absolutely. There's usually some in bars. Again, there was one in my uncle's house, so they might be in one of your family members' house. You never know. You're doing molecular phylogeny for these. So someone did, so that was one of the questions, if anyone's doing phylogeny. So for Largetooth right now, someone did back in 2014, they looked at their phylogeny, and they were all one species, and then they split, or no, they were all 3 species and they split, they put them all into one. But there's questions about that and what we might find from this particular study. might open up more questions about their phylogeny, and it hopefully will spark some others. So right now, no, but I think it will be something that people start doing after this. And they sequence the genome of the 5 species. They did the genome of smalltooth recently. I don't think they've done the others. They've done the mitogenome from the other species, so mitochondrial DNA. But the nuclear genome, I think the only one they've done so far is smalltooth. But that is something I think that they are working on.
Thank you, Annmarie. That was so interesting. Good work.
Thank you. Thanks for letting me talk about it. I love doing this stuff.
So I will process this recording and get it up on our YouTube channel sometime
Perfect,
in the
yeah.
next few days or so, and I'll send you a link.
Perfect. And yeah, if you get any inquiries, or I'll keep you updated. I believe, again, one of your guys' saws was a Largetooth. And I believe it's still, I think it's on my desk right now, so I haven't done another DNA extraction.
Keep us posted.
Yeah, we usually do one like once we get a pile of them. So
Sure.
once I extract it, then I will let you guys know how the DNA is and
Awesome.
if we got anything out of it.
Look forward to that. Thanks.
Yeah.
All right, last call for questions, anyone? All right, going once, going twice. Thank you so much, Annmarie. Appreciate your time and your expertise.
Yeah, thank
Pleasure.
you. Thank you, everyone.
Happy holidays, everyone.